Okay, so what I'm going to do, critical insects, and I'm not really talking, I don't know of any diseases that I'm really talking about, but I will be talking about some insect pests. So let's get started. I think the first one is what that I was asked to talk about was the codling moth and CideTrak CM. And the question was, does it work? And the answer is yes, it does work. It works pretty well. My research on this was, it was probably, what I found was that it's a version of something that's been around for a long time. They're using the pheromone, the sex attractant pheromone, and what they've done is they've microencapsulated the pheromone in PVC, in polyvinyl chloride. And so it's a slow release, and so it lasts longer, and you put out more of these into the field. Now, what I didn't find out, and which I probably is very important to you folks, is that does it work on small plot orchards? And that's always been the challenge with what CideTrak codling moth CM does. And those, all the products similar to that, because there's a bunch of them out there that are like that, and they've been out there a long time. So anyways, on this one, I just gave a little bit on the codling moth. Female adults about 3/8 of an inch long, and here you can see a picture of the adult. Very characteristic adult. Family Pterichidae, and all this family does. You see the way they curl their wings around like that in their resting stage. That's a pretty typical of that whole family. The larva is 1/2 to about 5/8 of an inch long at maturity, and it has a spot on the prothorax shield. I think usually you probably all know what the codling moth looks like. A couple of little things that I would like to go over. Overwinters as a larva under leaf litter and in loose bark on the tree itself or near the tree. And that's a little bit of a challenge for us, but it does overwinter as a larva. First flight, about first bloom, so it peak flight is about 2 weeks later after that. And then the newly hatched larvae bore through the fruit surface and generally calyx and feed near the surface for a time before boring into the core and feeding on the seeds and surrounding flesh until fully grown in about 3 or 4 weeks. So it's a very destructive pest. I will say this, that the west side does not have the best conditions right now for codling moth. On the east side of Washington, moth is native to Europe and was brought over many years ago and has always been a real problem for the Northeast fruit apple growing industry. But Eastern Washington is about the perfect conditions for this moth. We are not perfect, but we do get some pretty good infestations. It's not our main problem, but it's certainly one of them. At the orchard at the Evergreen farm, I actually, for training purposes, I really would like to have a bigger infestation than we get. But we usually get a couple of apples really that are going to be infested, so I can at least show them the damage and what the larva looks like. And oftentimes, because I put out pheromone traps, and I can show them the adult as well, and I can show them what the stings look like right there. So you can see there's one where the larva went into the apple early on. Here's an egg right there. There's a sting. Okay, let's get on to the next thing. 2 to 3 generations, that's in the mid-Atlantic region. We get about 2 to 3 generations as well here in the Pacific Northwest. And they can in Eastern Washington, they do cause significant problems. Okay, this is a really interesting paper I ran into as I was trying to write up this thing. Who actually preys on codling moth larvae? Because the biology of the codling moth, it makes it extremely challenging to try to kill it with pesticides of any kind, because the larva hatches, or the egg it will hatch from the egg, and then immediately that neonate larva then chews right into first it eats the eggshell in general, and then it will chew right into the apple and then head right in there. Once it's in the apple, it's kind of difficult to actually get to it with any kind of pesticide, because it's protected by the apple, and we can't use systemic pesticides on crops that we're going to sell to eat, because that pesticide then goes inside, it's in the fruit or in the product that we're selling, and people don't like that, and they shouldn't. But anyways, here's a really interesting study of some insects they looked at in terms of predation. And what this study was, and this is a really nice, I think, a breakthrough. Studying predators of any kind in a field, whether they're perennials or annuals, is a real challenge. The old way to do it was to spray the plant with rubidium chloride, which is a stable radioactive compound. It's a stable compound that does give off. It doesn't give off radioactivity, but it does. You can measure it. Okay. This new method is really neat and far more accurate. And what they do here is they collect these predators. And here you can see, here's those of you, you're probably familiar with all these. Here's an earwig. Here's a carabid beetle. This is a harvestman right here. These are not spiders. All right, this is not a spider. This is a harvestman, and they're a different order than spiders. And we know very little about them, by the way. And then here's a spider. And so these are the four main ones that they found to be preying on codling moth larvae and eggs, by the way. And the way they did this, and this is what really attracted me, was they removed the gut content of each of these insects. So they captured these insects, dissected them, and then they cleaned it up a bit, and then they did what's called polymerase chain reaction, which amplifies any DNA in there. And so what they're looking for is DNA of the codling moth inside the gut of these different predator insects. And they found them was in 8.9% of the 2,591 predators analyzed by this PCR product. And so here you can see here, these are insects. These are the different insects that were feeding on eggs and on the larvae. And so they're up there, and they can pull the larvae right out of the apple while it's developing and boring into the apple. Now you'll have an imperfect apple, but you'll have an apple that you can eat. And so these are earwigs, right? Earwigs. How many go around and kill their earwigs when they see them on the apple? Well, earwigs are a weird insect group, and they are omnivores. So they do eat plant material, but they also we've known that they are also predators. They also eat they're saprophytes. They eat everything, anything they can eat. So they're survivors, I guess you could call them. Carabids are ground beetles, but we know they do climb up trees and plants, and they will prey on eggs and larvae, depending on basically the size of the carabid. So there's a lot of species out there that are carabids. So leave those beetles alone. And then the harvestman, we don't know a lot about the biology of harvestmen, to be honest with you. There hasn't been a lot of studies. They sort of fall in the cracks, and people don't study them. So we knew they were somewhat predators. They're related to scorpions and whip scorpions. So we knew that some of these guys are actually very toxic in terms of their they have poisons that are fairly toxic, and they can some of the harvestmen that we find in the tropics can cause damage to humans. And then of course spiders here. So you can see here, and you've got ants and staphylinids. That's another type of beetle. These are the beetles, and you probably see them, and you'll see that they have little tiny elytra, so little tiny primary wings, first wings. and they're predators and parasitoids. They're a really interesting group as well. So these guys are going after it, and they are preying on it. None of them, I would say, okay, none of them you can count on in terms of controlling your problem. But it is interesting that the reason I put this up here is don't go kill these guys, okay? Leave them alone. Let them thrive in your orchard, okay? Anyway, that was, I thought, really interesting. But here's what you asked me to look at, and this is the CideTrak CM and how it works. And it works just like all the other pheromone release release products. And what you're doing is you're putting this product right up in here on the branch, and they're putting them up in the upper portion of the branch. And this is straight out of their advertising material. The thing with this product is that it's small and it's a slow release product. And so you're putting in a lot more of them. Now that's interesting because one of the challenges with this, the way this works is the male codling moth, the female codling moth doesn't fly. She stays put at the base of the tree through the winter, right? They pupate through they overwinter as a larva, then they'll pupate, and then they'll emerge as an adult. And the female just sits there and she's at the base of the tree. All right. And then she'll work her way up the tree. And she releases a pheromone. Now that pheromone, the male then picks up. And what she does is she releases a concentration gradient as she wafts it out of her abdomen. And what the male does is he flies in a sort of, back and forth. And what he's doing is he's cueing in through a concentration gradient of the pheromone right to the female and then mates with her. What these products do, and what this product does as well, is then it basically inundates your orchard, all right, with pheromone, and it hides this concentration gradient that the male needs to find the female, and now can't find the female. And so the female, she's gonna lay eggs whether she's mated or not. And that's what she does. She lays sterile eggs. Now, the problem with this product, and has always been, and this is on small orchards, because you have a large surface area, you have a large area around your border versus the mass inside. So as you get smaller and smaller, you have a larger surface area to your mass of your orchard. And so it's the edges of the orchard that really, it doesn't work well. So the more outside corners, outside area that you have relative to your whole orchard, the less effective these products are. Now, they didn't speak to this. I looked all over for this, but given that what this is doing is that it's a smaller dispenser, it's a slow release because it is microencapsulated, and you're putting out more of these products, these dispensers, through the orchard. Is it going to be more effective in a smaller orchard? I'm not sure, but it might be. It certainly does work. That was the question that was asked, and it works. It works well. It works as well as any of the others. I don't know that it works any better, but it does work pretty well. And you're applying to 10 to 20% of the total acre rate of the total acre rate, all orchard borders. So I'm thinking with this more concentrated, so in other words, you're releasing in more local areas, this could be more effective in terms of being used. All right, it could work. It could work on a smaller orchard basis. Worth a try. I wish I could tell you that it does or doesn't work, but I didn't find any research on that. I'm sorry. Okay. All right, that's you know what? There's no immediate questions on this. Okay, we can move on. All right, the next question, how do we distinguish between an apple maggot and a cherry magnet? Now, the apple maggot is our biggest challenge, right? That is our most detrimental pest we have in the West Side for growing, particularly for apples. And all of these, this whole family has they have ornate wings. Now, the difference, generally, you can tell to some degree, at least, you can eyeball the wings and you can say, okay, that is an apple maggot. So if you have an adult on a trap, you should be able to look at the wings and tell whether it's an apple maggot or not. So what I've done here is, here you can see, here's the apple maggot, and notice this brown streak. It's a reverse Z with another wing, another branch coming off of here. Now, if you compare that to the Western cherry fruit fly right here, or the cherry maggot right here, you can see that there's a gap right here. So the pattern on the wings are different and distinctive. Okay. Now, the cherry fruit fly and the walnut husk fly are more challenging, but there's a size differential there that should help you there. The walnut husk fly is much larger. Okay. And the black cherry fruit fly is also much larger and does have a general pattern a little bit more closer to the apple maggot, but it's not a terrible. It's distinctive enough that you could look at it and know that there's a difference. Okay, and this is a pretty consistent pattern. How do we monitor for them? Generally, for the apple maggot and for the Western cherry fruit fly, you can start out with yellow sticky traps. And that's because when the adult emerges, in the case of apple maggot, they emerge, sometime mid-June. I would start putting sticky traps out probably early June, mostly because of climate change. These insects we're starting to see some shifts, and they're emerging earlier and earlier. These are what are known as univoltine insects. So in other words, they have one generation per year. So you really have to deal with them once, but they can be pretty devastating. So you start out with a yellow sticky trap. And what happens there is, when the female first emerges from the ground, from the pupa, which they pupate in the soil, and I'll talk about that in just a moment, they are not gravid. All right. They do not have viable eggs yet. It's going to take about 10 days to do that. And they are not interested so much in mating at this point. What they are interested in is finding something to eat. And most of the time, they're very attracted to yellow at that point. Okay. And that's because a lot of flowers that they're interested in are yellow. And so they're looking for that. So the yellow flat sticky trap, sometimes some people will put a lure on there, a vinegar lure that draws them in. I've done both. I can't say that I've gotten any very strong distinction between any of them in terms of one working better than the other. We catch a fair number of apple maggots, but then the other trap that you need to put out is a red ball trap, and that after about 10 days. After about 10 days, that female becomes gravid, becomes reproductive. And now she is interested in finding an apple to lay an egg on. Okay, and so these red ball traps then become pretty attractive. I would suggest when you put them out at the same time, just to save yourself the labor and to remember to do it. And the other thing you can do with these guys is you can trap them out. That is, what it means is that you can actually use a trap to really impact their population. And this is, I'm not going on any published literature. I'm going on word of mouth from people, from West Side growers, and what they've told me: 3 traps per tree, and every tree, and you'll have an impact on the population. The other thing that also works, and I have done some research on this, is surround. They don't like surround. They're very much like every other fly, is that they have taste buds on their tarsi, on their feet. all right? And they like those feet and those tarsi to be clean, all right? And if you put surround on there, that clay material, that bothers the living daylights out of them. And they won't overposit if they have someplace else to go. So sometimes what works too is put a sacrifice tree out there and let them overposit on a tree that doesn't have surround, and they will go there and they'll just stay away from the surround. So that stuff works pretty well. And it's totally non-toxic. It's a little bit of a pain in the butt to get off the apple. And so you really got to scrub it off. And you have to spray it about every 10 days to 2 weeks. And you sort of spray it like you're painting. All right, you're going to put layer after layer, and that works. Now, there's one other thing I wanted to show you. And this is a new model, and maybe some of you already know about it. Kate, you may know about this, but this is the new degree day model that WSU has, and we used it in my class last year, and it's impressive. And I'm going to just jump over to that if I can, and I hopefully I'll be able to show this to you. If I can't, we'll go to it later. But let me try. Are you still with me? And so this is called Decision Aid System. And this is newly with WSU and with Ag WeatherNet. So the two of them are working together. This is, I believe, a private company in conjunction with WSU.
David, are you looking at something else on your computer right now?
Yeah, what are you seeing?
The same, you're distinguishing apple and cherry maggot slide.
Okay, so you're not seeing it. Okay, let's not do this right now. Let me come back to this, okay? Because I don't want to let me. I'm just going to go back to the PowerPoint if I could.
We're already looking at it.
Yeah, I'm okay. There it is. Okay, so I'll come back to that in a little bit, but remind me because it's really pretty neat. Also, by the way, spinosad also works on cherry fruit fly. It works on almost everything. It's a good compound, but you get resistance pretty quickly. Okay, we're back on this. Okay, now moving on to the next question. The pear sawfly, which I guess a lot of you have had problems with. Talk a little bit about the biology. So here's the adult. It's a typical looking sawfly, which is a hymenopter, which is a wasp-like creature, but this is a very primitive wasp. So the hymenopter are broken up into these more primitive, and then the bees and everything that you're more familiar with. Here you can see an egg right here on the upper surface of the leaf. In the biology and the life history of the pear sawfly, it overwinters as a pupa in a cocoon. Okay, and it's about 2 to 3 inches deep into the soil. Then the adult will emerge late April to May. And so you probably have noticed that and this is where degree day models come in so nicely, is that you'll probably notice that these guys are emerging. All of our insect pests are emerging a little bit earlier and earlier all the time, right? So you need to be on it. You can't wait to the last minute to do this, because everything's being pushed forward. Their life cycle is being affected by climate change as well. The female then inserts eggs into the leaf tissue, usually the upper leaf tissue. Egg hatches in about anywhere from 10 to 15 days. And then the larvae immediately begins to feed on the upper surface of the leaf. And you can see that here's it feeding. And it's a weird looking larva in this early stages. And so it feeds on for 3 or 4 weeks. And then falls off and pupates into the soil on the to pupate. Okay, that needs to be edited. Then the second generation will emerge in July, and larvae from that generation feed from August to September, and then most larvae from the generation drop off into the ground and overwinter in the ground. Okay. Management: you can hand pick the individual larvae, wash them off with a strong stream of water. And you can also, there are chemicals. Again, spinosad works, Kale and clay again works again. Again, these guys are getting, it interferes with their feeding. About 50 pounds per acre with the kaolin, okay? Be careful with the spinosad if you do decide to use it. I think the law says you can use it 5 times or 6 times a year because it does, resistance does happen pretty easily. Now, here you can see this is a typical larva of a sawfly. It looks like a lepidoptera, doesn't it? But they don't have prolegs, even though it sort of looks like it. They don't have. This is an older pear sawfly. Most of the time, they look like slugs, right? They're funny looking creatures. Here is one thing since I was an undergraduate, and when I was an undergraduate, I worked in an entomology department, and I always wondered about this. So many perennial tree pests, fall down into the drip line and overwinter or pupate in the soil. And right, and this is a natural thing that almost all insects do. They actually have a hormone that's secreted that causes them to what's called wander. All of a sudden, when they're ready to pupate, this hormone is released from, it's actually released from the brain, right at the side of the brain, near the brain, but it's produced in the very base of the abdomen, last abdominal segment of the abdomen. And it causes them to wander about. And here on this one, what it does, they fall off the tree. That's created by the hormone. They fall down and then they wander around into the soil until they bore into the soil, and then they pupate in the soil. And I was always wondering, why don't we put some kind of impenetrable cloth around the tree, right? A mulch, a plastic mulch, or any kind of weed barrier or whatever you want to use around the drip line, and so that they can't get into the soil. Now they'll still pupate. All right. This actually, if you don't do anything, they'll live unless a predator finds them, which they are very susceptible to that because they're sitting right on the surface. But you can also bring chickens in or something into your orchard, and then let them go after them and feed on them. They will love them. And this is actually, you'll see when we get to Drosophila, you'll see actually that is a strategy now they're starting to look at with Drosophila. But even as all the way from undergraduate, I thought, why don't people do that? And I still don't know the answer to that. And it could even be a temporary mulch. So if you're really into Michael Phelps, nutrient management programs where, he's using those little tiny wood chips to use it as a nutrient management, you can then just save your drip covers, these covers, and just put them on when you need them, and then take them off at the end of the year. And you could still use that Michael Phelps, that wood, that chopped up wood as your source of nutrients. Anyway, it's just a thought. I don't know if it would work. I think it would work. I don't know why it wouldn't. Even if as you get dirt on the over that barrier, over that mulch, that plastic mulch, it's still, the chickens will find a lot of them. Anyway, so that's it, and we can come back to this again too if you want to turn. All right, now the next one that I was asked about was the life cycle of the spotted lanternfly. Do we have one? I think the question was, do we have it in Washington? All right, the answer is we probably do. And I will show you some models of that. But we probably don't have huge populations of it yet. It is a major pest. And I think it is over time, given some time, we will have it under control. I don't think it'll be super serious, although if it starts transmitting disease, I will take all that back. Okay, so here it is It is a hemipteran. It is a plant bug, essentially, is what it is, and we have a lot of plant bugs. The damage that it causes, it chews into the stem and branches of the plant and sucks out sap. So very similar to any of the plant hoppers that you've seen, or leaf hoppers or tree hoppers that you've seen. And these guys, these fulgorids, this family has very ornate pronotum and things like that. And that's these guys. This is sort of a typical looking fulgorid. It's out of Asia. It will cause wilting. It'll cause leaf curl. It'll cause dieback. It also excretes a honeydew, just like aphids do, and will cause a sooty mold, which then will cause problems with your photosynthesis and other issues. So it is a pest, all right, for sure, and it is a problem. And especially, apparently, the numbers can be pretty significant once it establishes itself. It was found, I think, in 2008, I want to say, in Pennsylvania, and has moved from there. And I have pictures of the immature stage. Here's an early instar. This is the adult. Here you can see they're quite dramatic and rather impressive looking insects. Here you can see an egg case. The eggs are kind of hard to find there, but you can see it. It almost looks like a stream of mud coming out, but they usually lay eggs in groups of about 50. And so where do we start? Let's start. So the eggs will hatch first instars in May and June, and you can see they're bright red. All right, so that takes a lot of predators. Predators will be warned off from that, the general predators. Second instar is June to July. This is New York, by the way. So, and they're hotter than us. Okay. So this probably is a little bit sped up for us. All right, third instar June will be June, July. Fourth instar July through September, and the adult appears July and December. And then they lay eggs from September to November, and you can see in that little tiny picture, you can see the eggs. One of the things they're doing, because they are crawling, you can see most of the pests are non-flying. They don't have the capacity to fly here, the immature, because they don't have wings. And so these sticky bands, because they'll crawl up the tree, can be pretty effective. All right. Biocontrol are predators, parasitoids, and entomopathogenic fungi like Bulgaria and things like that can work pretty well on these guys. Predators and parasitoids are coming. They're all in Asia. They do a really good job. These are not a pest in Asia. Okay. All right. You want to also destroy the egg masses. And the way we will get this is not for them flying over across the country to get here. We're going to transport them. Okay. Because the egg masses look like just a little stream of mud that people will ignore, and they will drive them in here with your vehicles. All right. But if we can destroy the egg masses, then we can keep them under control. Insecticides labeled for plant hoppers, and they do have some systemics there. Again, you really can't use them on trees, so you can use trap trees, and trap them out. So, anyways, they're working on lures and trap types, and so I do believe that this will eventually get here. If it's not already here, it's here, and it probably is here in low numbers. But I don't think it'll be as serious of a problem as some may be worried about. Okay, so here's its range. Right now, here it is. This is where it's really a problem, and here in the middle Atlantic states here, and in the Midwest, Ohio. So you can see it here. It's really in New Jersey, Pennsylvania, and into New York, Connecticut. Again, this is where they've been identified and clearly have them, there is no question. But these maps are very conservative. Okay, so if you're finding it here and you're finding it here in this county, in Pennsylvania, they're in all the counties. Okay, they're probably all over. All right, so they're probably within this whole range here. They're probably in eastern Ohio, into West Virginia and Virginia. Okay, and may or may not be in North Carolina. All right, here you can, and probably are though, because actually they are. Here you can see a more blown up model. We have it in Oregon. They've been clearly identified. And one of the areas that is considered a problem area is in the Yakima area. Again, they want some serious heat, which we don't have yet. We are in an area that is a fairly low risk area. The Yakima County area is a high risk because they have high temperatures, which they like, lots of sunlight, and lots of trees, lots of fruit trees. Okay, so that's where you'll see the problem. Again, here you can see Central Valley, San Joaquin Valley in California, same deal as Yakima. They got a lot of heat and they got a lot of things for them to feed on. Okay, so you can hear same thing here. You can see these are hot, humid areas. They do like humidity. And so that's gonna probably keep it a little bit down a bit. Here's where they've been identified. You can see it in Oregon. I looked at it and I have not seen it it has not been identified, at least, officially in Washington State, but I wouldn't be surprised. Okay, and so this is the potential area, and this down here is the area itself that we now know where they are. This is the infestation. This is identifying where we've actually officially identified them. Okay, so that's the distinction of these three maps. Okay, so that's it for them. How are we doing time-wise? How am I doing time-wise? Anybody? Okay or not okay?
I think you're doing, as far as I know, I mean, I don't know how much further you have to go, but yeah.
I don't either. I got 20 slides, and so I'm close. I'm almost there. I'm gonna have a little bit more on the spotted wing Drosophila, and because that's a problem we do have, and again, very similar to the lanternfly, but this guy was first identified in California in, I think, 2009. And suddenly in 2000, by the end of 2009, it was in Oregon. And I think by 2010, it seemed like it was everywhere. So this guy has flown everywhere. And because of our Himalayan blackberries on the west side, we thought we were going to be overwhelmed. And I don't think we have been overwhelmed. I think we've had some serious problems with this pest, but it's not what I thought we were going to have. I really thought, oh man, this is bad. But $718 million. Yeah, that's kind of chump change, but it's not. That's a lot of money. Management costs are around 129 million. And we've been kind of winging it on this, and I think done a reasonably good job. Because I mean, it was all hands on deck when this thing showed up, and they did a great. I think you can see how many schools are involved and research institutions. There's a lot of, and there's more than this, but there's a lot of them, and they've gotten into consortiums and things like that. So they've worked well together. So here it is. So let's go from there. Eggs. This is a Drosophila. So any of you remember from high school genetics, you raised Drosophila in these little bottles, right? And they grow fast, right? These guys are very similar. That was Drosophila melanogaster. This is Drosophila suzuki. So this is a group, what is called the vinegar flies. They're not fruit flies, they're vinegar flies. They really like rotting fruit. All right, and so in the lab, you fed them a banana extract in agar, and they loved it. In the field, you have lots of Drosophila out there, not just Suzuki, and they're feeding on the rotting fruit that falls off the tree. And they're totally harmless. In fact, I don't know if they're doing you a favor or not, but they're certainly not causing you any problems. But this little ovipositor here's a female ovipositor. And you can see how it's serrated. And this guy has made the transition from rotting fruit into live fruit, healthy fruit, and ripe fruit. They like it to be ripe. Okay, it lays an egg right in here. And then it has a little breathing tube. So it actually has adapted. It's a really, evolutionarily speaking, this is actually a fascinating little creature because it's made these transitions and these challenges that feeding on rotting fruit, you don't have. But when you get into ripe fruit, you have some problems. How does the larva breathe? And it has a little breathing tube. And so it goes in just on the inner surface of the fruit, and it feeds on that, and usually has a little tube where it can breathe out of on the surface, and it feeds there on the tissue, and that does that for 3 to 13 days, and then it pupates. It can either pupate in the fruit that falls off, or it can pupate. It can come out of the fruit and pupate into the soil right next to it. 3 to 15 days later, again temperature dependent, the adult comes out. The male, this is where it gets its name, the spotted wing. Here's the spot, and this is the male. The female, you can see, doesn't have the spot. All right, and so this is actually, do you have spotted wing Drosophila? That is the challenge, and so you have to do some extra work to actually know that. Although I'm going to show you some stuff that they're really making some nice breakthroughs on that will save you, I think, a lot of problems. So let's go on to the next one. There we go. Okay, this is its range. 2008, August 2008 in Santa Cruz County, they first identified it. All right, and you can see it's spread all over very quickly. It was in Oregon and Washington State. It was probably in Nevada, but they didn't find it. But you can see it was in Utah in 2010. And then it just worked its way over. So it got all the way over here into the Carolinas, Florida, and it also into Mississippi. And so that was probably human transport that we did that for them. And so you can see the ranges. They're pretty much everywhere. This is the really Midwest in here, where it gets really hot and dry, hot and humid in the summer. They're not really crazy about that. They don't like really high heat. They really like it to be a little bit cooler and humid. And so what we have found, and they have been, and you can see here in Hawaii, they've been there since 1980. Okay, so that was actually a real benefit to us because some of the predators were already brought over from Asia. This is another Asian pest, by the way. Okay, what else? Is there anything else on this slide? No. Okay, let's talk a little bit about some of the strategies that are taken. This is a very cool strategy. It's called Splat technology, and it's specialized pheromone and lure application technology incorporated with a pesticide. And so what this test was doing, what they were looking at is some various versions of this Splat technology. This first one is spotted wing Drosophila. They have a red dye and spinosity. So they have, this is an attractant, a pheromone. And then they have the second one. They have 8 or 9 components that act as an attractant blend. And then finally, here is another one that has 4 components as an attractant blend. So, and you can see it, I showed it here easier, but this is supposed to show it right in here too, that what they're doing is, and you can see the red Drosophila loves red. I had a student last year, she did a study. We get Drosophila problems late in the season. So the end of our raspberry harvest, we start to see some serious Drosophila problems. Early on, not so much of a problem, but we do take precautions anyway, because if you want to chase a customer away, put a maggot in their fruit that they've eaten partially, and that does a really good job of getting rid of your customer. Anyway, so let's talk about what this technology is. So what it is this is it. It's usually in some kind of a paste or a glop. All right. So you put a dollop on your tree or your bush, and in that dollop has some kind of an attractant, usually some kind of semiochemical. And that is a chemical that is attracting the insect to the plant. So you're trying to get the insect to come to the plant. All right. And they will come to the bait. And so they're attracted to this. They're going to ingest some of this solution because you're also, usually it has what's called a phagostimulant. That is, it encourages them to eat a little bit. Okay. And then what you have in there is some kind of a pesticide in general. In conventional, it's some kind of pyrethroid. This group, I think, mostly use spinosad. But I think as they get more sophisticated, they could be using some viruses and some other biocontrol agents that might be effective. So anyways, that's how this works. And this is a technology that is gotten very popular over the last, I would say 5 to 10 years, where you're really, instead of spraying your field, you're putting out these little dollops of pesticide, and you're really just, you're using it. It's on a little, usually a spray can or something, and you just spray it in 1 or 2 areas on the tree, or maybe 3 or 4, but you're really concentrating where it is. And then if you're using a toxic chemical, which conventional growers would, you're really isolating the use of that pesticide, and it's not spreading everywhere. So you really cut down on how much you use. And if that attractant is really effective and the phagostimulant is effective, and that usually is, this is a really pretty effective tool. And organic growers now are using organically certified compounds that will work. And here you can see it. And this is really some mortality data that they came up with. And you can see here, these lower bar graphs over here are just the attractant and the dye. Okay, no pesticide. Okay, and you can see, look at 40%. That's I wouldn't. That's not bad. That's okay. I'm okay with that. But then as soon as you add in truss, which is spinosad, you can see now you're getting some pretty high numbers. All right. And this is just, these are the days of a sampling that they did. Okay, so it's very effective. Okay, so this is something, I don't know that these are generally available yet, but I think they will be in the near future. Now, this is really exciting too. These are some using augmentation. None of the native predators or parasitoids are highly effective, but right now these are introduced and they are only being used now really on a research basis, but they're working and they're pretty good. And you can see this is a pupal parasitoid. This one, I don't remember what this one is, but these 3 parasitoids. They all look like they're chalcids in the family Chalcidae. They're still looking. Okay, this is definitely in the research phase. Okay, but I think you're going to find, once they start really searching in throughout, it's native to central China, Mongolia, and just throughout in Korea. I think it's also in Korea as well. So I think they'll find what they need out there eventually. It's going to take time because then it has to be quarantined and shown effective. So anyways, biological control really works. Augmentation with native parasitoids, increased parasitism, but it's not effective in reducing the numbers in the fruit infestation yet. Some of these exotics from China and from Asia in general are showing more promise, which makes sense. Entrust really, honestly, if you look at the data, Entrust is the only one that really works. I wouldn't mess with Pygamic. It doesn't really work all that great. You're just spraying it out there. And these other two also aren't all that great. So I think this is really has some really good results. I think I have one more on this. Oh, here, this is the one I wanted to do. All right, this is the one I'm most interested in. So monitor your field with traps. So anyways, I think I started to talk about this. One of the things one of my students did, she took the apple maggot red ball traps. And what she had to do is she really had to slop on the sticky trap on there. And really, so it's thick. And then she put those out and they were really effective on collecting spotted winged Drosophilas. So if you have those red ball traps and you want to get rid of spotted winged Drosophila, it can work. But anyways, the trapping is usually a yeast baited version of it. And it's a good trap, but you have a lot of Drosophila and they're all attracted to this. Okay. So you're going to have to sort them and make sure you can identify spotted winged Drosophila. That's probably the biggest challenge with it. Right now, people have tried to use trapping out with this. It doesn't really work all that great. So another thing they've tried to do is what's called exclusion netting. So grow your whoops, I didn't mean to do that. Grow your crops within an exclusion area in a high tunnel type situation, and yeah, that works really good. There's other challenges with that, right? Especially with heat and everything else. Here is something I think is really interesting. So everyone, Michigan, Georgia was involved with this. I want to say North Carolina, NC State was involved with this. So California was involved with this at Berkeley. Oregon State is involved. WSU wasn't involved. Michigan State is involved. So the whole country, every one of them were saying that early in the season, every growing season, spotted winged Drosophila is not a big problem. It builds up through the year. And that's what we have found at the organic farm at Evergreen, right? We don't really have a huge problem early in the season. It's later on. It's in the fall, late fall. Then we got Drosophila everywhere. But early on, we see it, but we don't have a lot of it. All right. And so what they're doing now is get some early maturing cultivars. And strawberries are great. June-bearing strawberries are really great. They don't have much of a problem at all. It's ever-bearing ones that do have some problem later on in the year. Do some pruning and mulching and exclusion netting, sanitation, and harvest frequently. Now the mulching, one thing again, these guys are pupating in the soil, and this is what they were doing. They were putting plastic mulch down. Okay. And so they were able then to keep the pupa on the surface, and they're far more susceptible, and you can then use sanitation and clean them up yourself. And that really works. All right. And so even though we don't have a biological control agent yet, you can see now, I think we have a reasonable strategy that you can produce good fruit and not be overwhelmed with spotted winged Drosophila. Also, entrust works. Entrust is tough on honeybees, by the way, so keep that in mind. And it also, resistance has really built up really fast with this thing. But, so make insecticide applications early in the morning or late in the evening to target peak spotted winged Drosophila. Really early in the morning. Make sure honeybees aren't out, okay? Now here's one that we've been doing at the farm. Okay, so for our mostly raspberries and strawberries hasn't been as big a problem, but raspberries have been somewhat of a problem. We harvest 3 days early. All right, we put them in at 35 degrees Fahrenheit for 3 days, and everything is killed. Even if there's a larva in there, if it's not wiggling around, more than likely nobody's going to see it, and it tastes like the raspberry. Okay, you got a little bit more protein. That's it. So I don't feel bad about that. Um, but 35 degrees for 3 days will kill close to 100% of everything. Okay, that is your probably right now your easiest task. If you're growing them at home, take them, harvest them, put them in your refrigerator. That's going to be 40 degrees. So you might want to keep them there a little longer. Or 37, right? I think the refrigerator is supposed to be 37, something like that, somewhere in there. And you should not have a problem with them. Okay. But I think that's a great strategy. And then of course, early cultivars, start looking at early maturing cultivars, and get in and out of there before they get to be a serious problem. Okay, so that's where we're at with spotted wing Drosophila. They're making great progress. I saw a webinar on the latest research, and I was really impressed. They're really very coordinated and doing a lot of really nice work. All right, climate change. Okay. Any non-believers here? Climate change is real, and it's if we don't do something, it's going to do us in. I'll be honest with you. We are seeing serious effects already. We're seeing temperature increases. This summer is supposed to be hot and dry. I mean, we already have a pretty dry summer, but it's going to get hotter and drier. We are actually in a little island. We're very lucky in Western Washington in that our impacts are going to be lighter because we will have more rain. We're going to get wetter first before we start really drying out. So we're going to see temperature increases, which we are seeing. Longer growing season, less frost, warmer nights. Okay, insects absolutely love that. That's great. Diseases also love that as well. Precipitation changes. We're probably in many cases, at least early on in this climate change, what we'll see is that maybe our totals will look exactly like they always have in terms of total rainfall in certain aspects, certain parts of like Western Washington. But what we'll see is the pattern will change. Instead of that light Pacific Northwest rain that just never stops, it's going to stop, and it's going to really rain harder and harder. And you're seeing this in other parts of the country where they're drying out, but at the same time, they're getting these superstorms. And that is exactly what we were predicting. So mixing rain and snow. So the weather is going to just be less predictable. And increased intensity of precipitation events, more flooding, more droughts. That is really the big one. Increase in carbon dioxide concentrations. This is an interesting story. So I was listening to a podcast from Oklahoma State University, and a postdoc was working on the grasshoppers of the prairies in Oklahoma. And what they're finding in the spring, conditions look great. Grass is growing faster than it's ever grown before. All right, it's growing super fast and high carbon dioxide. All right, and warmer temperatures. All right, and they're growing fast. But what they found is the grasshopper populations were actually collapsing. And the reason for that is that you haven't added any new nutrients to that pasture. And so what you have is more biomass, but the nutrients, basically you're just diluting out. This is a low, sub-nutrient quality. The nutrient quality of your plant has diminished because it's sort of like growing crops with urea. All right, they start to grow really fast. You inundate it with high nitrogen early on, and nutrients can't keep up. I don't know what that was about. Sorry. Okay. I just got a couple more. Okay, effects and sensitivity to varying commodities. Corn, high nighttime temperatures, high temperatures during pollination, and water stress. All right, so pollination is going to be affected. All right, soybeans will be water stressed and high temperatures. They don't like that very much. Wheat and small grains, you're going to see extreme events. All right, frost during flowering, water stress. All right, so again, all of these are telling us that our yields are gonna go down. Okay, and and I don't anyway. Rice temperature extremes during pollination, water manage, and you're going to need to be more water management. Cotton, high temperatures during boll fill. All right, that's a problem. So you're not going to get is full bolls. All right, and so your cotton will be your harvest will be less. Pastures and rangeland, just water stressed. All right, because it's hard to water them. Fruit trees, sometimes, in many cases, chilling requirements are not going to be met. Now we can do some genetics on that, and we can solve that problem, but that's going to limit you. Most of your higher quality traditional fruit, your heritage fruit usually has a chilling requirement. High temps during fruit development usually means there's going to be a lot more fruit aborted. Specialty crops, a lot of water stress, high temperatures. There's going to be some compromises. Where are we going to get the water? All right, because everybody else wants it too, and there's going to be less of it. Okay, some more things. Climate change, direct effects. We're going to actually see morphological changes in many of the plants we grow. Physiological changes, because we're going to see increased temperatures, then decreased temperatures. So we're going to be seeing more variable temperatures, phenotypic changes on them. Also, basically, it's all impacting plant productivity is going to be reduced. Indirect effects, we're going to see soil fertility. We're going to see nutrient cycling is going to be badly affected by these high temperatures, then low temperatures. There's certainly some, but mostly high temperatures and dry conditions, that's going to impact soil nutrient cycling. Irrigation availability, more irrigation. All right. When I was working in the Southeast, when I was younger, nobody irrigated. Nobody had irrigation equipment. And basically the rule of thumb was if you're east of the Mississippi, you don't need to irrigate. Well, that's not true anymore at all. All right. Everybody's irrigating. Rise in sea level. All right. And then also salt infiltration with that rise in sea level. Increased pest numbers, because we're going to see longer growing seasons, hotter temperatures, more pests. Heat, flood, and drought. All right, we're going to see all of those. By the way, pests, every generation is an exponential growth. So if you, instead of 2 generations, now you have 3 generations, it's not just a little bit more, it's a 10-fold more. Okay. Now, socioeconomic impacts, food damage, demand is going to continue to go up. Farmers are going to have to respond. Costs are going to go up. Policies are going to have to be modified to address that. Will there be trade? I bet trade gets interrupted. I'm not even talking about human migration as food sources dry up, particularly in underdeveloped nations. People are going to migrate away from the Ganges in 2024. The glacier that feeds the Ganges River is going to be gone. Okay, so that river then will become intermittent. All right, that means everybody that's being fed off the Ganges is going to have to go someplace else to get food. All right, so it's where are they going? All right, it's it could be a mess. Okay, so human intervention, adaptation strategies, and mitigation strategies. We can do some of it. We can't do all of it, but we can do some. But we can, certainly if we can just keep this at a reasonable level, we can deal with these changes. But once they get out of hand, then they become more difficult. Well, this is my last slide. Some positive effects. All right, increased productivity from warmer temperatures. All right. Decreased moisture stress. Okay, so some diseases will probably be less of a problem. Possibility of growing new crops in your region. All right, if you're a wine grower or growing grapes for wine, you're gonna probably do okay. Grapes are probably gonna be one of the few crops that really is pretty good for climate change, at least for a while. All right, accelerated maturation rates. A longer growing season, increased productivity from the enhanced CO2. And I've already talked about that. That's definitely, that's a totally mixed bag there. And here you can see the negative effects. Crop damage from extreme heat. Planting problems due to the less reliable forecasts, all right? Because the weather is going to be more unpredictable and more erratic and more extreme. Increased insect infestations, definitely. Huge rainstorms, big rainstorms that actually cause damage, all right? Rain and hail, all right? So it'll be more dramatic weather. Increased droughts, all right? Increased weed growth, because weeds love this stuff. This is great. They're right there. They're ready to go. Increased moisture stress, which means you're going to have to irrigate more. Increased crop diseases in some cases. All right. Increased ground level, ozone toxins, green plants. So again, more pollutants, stronger storms, floods, warming stress, waterlogged land, more soil erosion, specialized monocrop, less adaptable. All right, so we've got to really get away from monocropping, and I think we will, because that's going to be a failure. Sickness, loss of human labor. These, you got to have laborers out there, people working. I already have it with my students, right? When we had those fires last year, not last year, but the year before, right? The students couldn't go out for a week. And then, so if they do, they have to wear a mask or something because the smoke is so strong. All right. That's a real problem. That's real. Right. And so if you've got laborers out in the field, they've got to deal with that in some way. All right. In some cases, maybe it's just not possible to do this kind of physical labor. Decreased pesticide and herbicide efficacy. We're going to have to come up with better strategies. Okay, on that high note, I think that's all I got. Yep, that's it.
David, you wanted us to remind you to circle back to the degree day modeling.
Oh, right, thank you. Yeah, I think you'll find this pretty neat. Can you see this?
No. Not yet. I think you'll have to.
I have to go to, hold on. Where are you? Oh, here. Yeah, there it is. I got it. Okay, good. Okay. Now you can see this?
Yes.
Okay, so this is, and I've already signed in, so you have to have a password. I think they do charge. They gave this account to me just for out of their totally kindness, so that I could use it for my students. And so this is called the Decision Aid System, and it's through WSU, and AgWeatherNet is doing this, and it's part private and again part state run. Let's see, where can we go? Let's go my stations. Okay, so the ones I used were Pasco and Tumwater, and I used that because it was Pasco and Tumwater were just two extremes that the students could see differences in the east and the west side in growing. So let's see, let's do, okay, edit. Wait a minute, let's go. Pesticide effective, maximum residue. View.
Jeff?
Sorry?
Are you watching something with sound?
Sorry?
What am I hearing?
I don't think we're hearing that. I think it might just be you.
I'm sorry. My apology.
No problem.
Sorry.
Okay, so here's what we have here. Here you can see here. So here's Tumwater, which is what we used because it's the closest AgWeatherNet station that we have to the organic farm. And here are all the different pests you can look at, including apple scab. You can look at apple maggots. There's fire blight. Somebody asked me about fire blight. Shoot, I meant to put that in there. Why isn't that in there? Anyways, fire blight is not a major problem on the west side yet. Anybody have fire blight issues? Because I don't think somebody does.
Oh, I think, yes, like pear trellis rust,
Pear trellis rust, yes. Do you have fire blight?
well, I don't know, I guess.
Well, right now we can get it here, but we're really not. It's not like Eastern Washington, and the model will kind of shows you that. But let's look at apple maggot, because that's coming. Oh, it hasn't started yet. So let's try codling moth, because they're right there. We're here with codling moth. And so we added a new set of charts to show you effectively. Okay, so here's what here's what you get. And so today we're at 176 degree days in Tumwater. And then you can see here's the prediction for the next few days. Degree days on May 4th. We're going to see 100. We're going to see 176. Conditions: 1% of codling moth adults should have emerged. No eggs have hatched, but they should be starting to be laid at 225 degree days. So next week. All right. So in about 14 days, on May 18th, they're predicting that we're going to start to see we're going to start to see eggs hatching. So they're recommending organic management. Mating disruption is a key component of the codling moth management in organic management orchards. All right, so you want to have those out there now. You should have had them out there a little earlier. And let's see, what else do they say? Mating disruption is key component. More effective, particularly high pressure situations, pheromone. So that's really all they're saying. If mating disruption dispenses have not been applied, do so soon as possible, and also apply an oil application at 375 degree days to kill any codling moth eggs that have been deposited before the dispenser application. Okay, and does everybody know what degree days are? Anybody not sure what degree days are?
No, I am not sure what they are.
Okay, so what we do here on degree days, so insects, plants, lizards, a lot of animals don't control their temperature. They're not what are known as homotherms. All right, they're pokemotherms. Their temperature, their body temperature is essentially whatever the ambient temperature is. Okay, and so their development is dictated by whatever the temperature is, because their body temperature is dictating how fast all the metabolism is taking place and how fast they grow. All right, and so we can then measure the average temperature. All right, so we take the high temperature. This is the traditional way of doing it. There's more sophisticated ways to do it. You take the high temperature and you take the low temperature, and then you take the average. And then you have a threshold somewhere in there where below the temperature insect isn't affected. But once there's a number of degrees, an amount of time above that threshold level, then the insect is developing. And then we can measure the number of hours at that above that threshold temperature. We can then predict what life stage the insect is in. When is it going to be an adult? All right. If we follow the temperatures above a certain threshold, 45 degrees is usually Fahrenheit is usually that threshold. Anything above that, all right. If the temperature, as long as it is during the day above that temperature, during that whole period, the insect is developing. All right. And all we have to do is measure how much of that time the insect is spending in the temperature above the threshold. And then we can then predict where that insect is in its development stage. It starts out as a larva in the winter. It's sitting there because it's generally below 45 degrees. But once we start getting above it, it starts to develop. And once it gets to so many hours at above 45 degrees, suddenly it's a pupa. And then we get a few more hours above that 45 degrees, and then now it's an adult. Once it gets a few more hours above that 45 degrees, now it's starting to lay eggs because it's gravid, okay, and fecund, okay? and so on and so forth. So we can follow it using these models, just following the temperatures through and you can do it. And the way they used to do it is because you remember those thermometers in the greenhouse where they give you the high and the low for the day, right? That's where that comes from. And so what they would do is they take the high, they take the low, and take the average, and then they would put a threshold. Anything above that, all right, the thing was developing. Now you can use more sophisticated techniques and say, okay, here's how many hours it was above that temperature. Does that make sense?
Yes, it does. Thank you.
Yeah.
So is that 45-degree threshold, is that the most common base
Yeah,
temperature?
for insects, that is the most common. It is pretty much. I mean, it's going to vary, and they can measure it from insect to insect, and that's certainly that's work. That's somebody doing a master's degree. That would be a nice little project for them to do. You got to just have a growth chamber, and you can figure that out.
Okay, interesting. So someone asked that question in the chat.
Oh, okay. Yes, that's right. That 45 is pretty typical. But it is it is species specific. So here you can see the cumulative percent development versus degree days. And then you have to match up that development with the model. And then what they're doing here is the flight chart. So they're actually following the adults, and traditionally they start this model once they've captured adult codling moths in traps, and they have a steady collection of traps. Then the degree day model starts. Now they just started from the beginning of the January 1.
Okay.
And
interesting.
Here's some raw data.
All right. So this tool is available only to people who request access, or?
Yeah, it is
Not to the
actually.
general
And your county extension agent, if they're an ag person. Kate, you're familiar with this, correct? Kate, are you there?
Yes. Sorry, I had it turned off. Actually, no, I am not familiar with it. So I was looking at it while you were talking, and I must have triggered something because I wanted to look at two different stations, one here and one in Yakima, and it wants me to spend $440.
Oh, wow. Well, it's gone up in price then. Well, that's a problem. I'm sorry, I didn't know it was that expensive, because like I said, they were really nice. They were wonderful, and they were to me, and they let us have two stations to look at. Wow, $440. That's a lot. That's too bad, because
$220 a station.
Oh, that's really too bad. Because it is a really neat tool. Look at it, and the problem is they gotta pay for it one way or another. Somebody's got to pay for it. So I'm sorry. I was. Let's see what else we can do. Natural enemy.
That's all right. I think it's very interesting that they even have the tool.
Yeah, they do. This is Wenatchee is really, in terms of a codling moth degree day model, they have one of maybe the best model for codling moth in the world.
Okay, great. I just want to get to some of our questions in the chat, and
yeah, please go ahead.
If anybody wants to ask their own questions. So someone asked about the apple maggot traps. What goes into those traps?
Oh, which traps which ones are we talking about?
For the apple maggot.
Yeah, there's two traps for the apple maggot. There's a yellow sticky trap, and that's just a yellow. I'm trying to get out of. Hold on. So I can see you guys. Oh, here we go. Stop sharing. Okay. So the red ball trap. Both of them have you can get a vinegar lure with them. There's no pheromone on this. So this is just a visual trap. And the first one is the yellow sticky trap. And then you use sticky tack trap. And you can buy that. There's Great Lakes IPM is a great place if you want to go there. They have lots and lots of stuff, and you can get it in a spray can because it is a messy product to deal with, but it generally comes in a paint can, and you can paint it on. but you're then gonna have to take a bath afterwards. And you know, at Evergreen, we don't really like to do that. But you can spray it on and it works great. So that's really all there is. So there's a yellow trap, and then there's a plastic ball trap, and it's a red, it looks like an apple. And then what we do is they're both made out of plastic, and so you can reuse them over and over again. They're not expensive, but there's no point in buying them if you don't need to. And you just take a paint scraper and you just scrape off the insects in the sticky trap and then reapply, and you can use them for a long time. They last.
Oh, good. Okay, thank you. And someone asked, what is a trap tree? You'd mentioned that as a strategy.
Okay, a trap tree is a tree that you're untreated. You're gonna, it's sort of a sacrifice.
Okay.
Food.
Someone asked about covering the ground with cardboard and mulch, if that would deter the ground.
Yeah, you know, right, absolutely. I think that would work. You know, I haven't been in the primary literature, but it's one, you know, let's think about it from the insect's perspective. They fall out of, they climb out of the fruit and fall to the ground, or the fruit falls to the ground and then they climb out of the fruit on the ground. And then what they do is they wander about very slowly until they find a spot, and then they just bore down. And there's nothing special about the spot they're finding. They just wander for a certain amount of time as they have that hormone going through their blood system. And then they bore down into the ground, and then they pupate. Now, if we put a physical barrier there, right, they're going to have a hard time doing that. They're going to be on the surface. Now, then from there, you can just, you can even put it underneath. So if the cardboard, and then you know, at the end of the season, pick it up, and depending on the species and the life cycle of the insect you're trying to control, you should be able to pick it up and find those insects, or feed them to your chickens.
Yeah, the person who asked this mentioned cardboard and mulch. So I'm not sure if what kind of mulch they were.
Yeah, maybe wood chips or just a compost mulch. And it'll make it a little bit more difficult for you, but it still would work. You at the end of the season, you'll want to pick it up or whatever. Again, depending on the life cycle of the insect and when you're doing that, you should be able to find them pretty easy. You know, just pick up the whole thing with the mulch and the cardboard and put it in a wheelbarrow if you want, and then you should be able to filter through that, or even through a screen filter, you know, screen system.
Okay, excellent. Thank you.
Yeah.
Someone mentioned that there have been some degree day models that are free using local weather stations hosted by Oregon State with a wide range of models.
You know, yes. So AgWeatherNet will have some degree day models. So if you go to AgWeatherNet, WSU AgWeatherNet, and that is free. So you just have to register, and there are models there. And Oregon State does also have a codling moth model. You're right, they do. Depending on where you are, but AgWeatherNet is very east-centric, Eastern Washington centric, but they do have stations. But they have a station on Vashon. They have a station here, 2 stations here, East Olympia and in Tumwater. 21 Acres in Woodinville has one. I know that the San Juans have a number of stations, and they'll show you where all the stations are. They have a map. That's what you open up to. So you go to WSU AG Ag Weather Net. That's one word. And you'll find that, and then they'll ask you to register, and then you'll see all that, and they do have that. They just don't have all the other IPM tools that this decision system has, which is.
Okay. Well, that's good to know. Thank you.
Yeah, and yeah, I'm really sorry. I hate to hear that it's so expensive. That's unfortunate, but you know, they need to survive, and we want them to survive.
So there's a question that just came up in the chat. Are there any concerns about importing more non-native bugs to be used as biological controls, because it seems like some of these approaches have backfired?
Always, yes. And so in every land-grant college that I know of has an entomology department, has a very sophisticated quarantine system, a building that is just for quarantine. And it's a negative pressure system, double doors. It's really because that is exactly right. We have early on, we had a couple of successes and we had a lot of failures, and really bad failures. And so, yeah, every one of them does. So they quarantine them. So entomologists will go out and they'll be in China. For example, with the fire ant, they're still looking for predators. That came out of Brazil. So people go down to Brazil and they go to areas where the fire ant is just in very low populations. And they're looking for why are they at a low population? And it's usually because they've got predators and parasitoids that are keeping them at that level. And so they try to find them. Then they bring them back. They put them into it. They develop a culture, and then they test them against various insects, beneficials and pests, and to see if this thing is going to turn into a pest on itself. So yeah, that is a major concern.
Okay, yeah, thank you for elaborating on that. I had a question. Actually, I've subscribed to some industry newsletters, and I read an article about a company that's maybe trialing the sterile male technique to control spotted wing Drosophila, and I was wondering if you know anything about that. And
No, I haven't seen that one. That one, that's an old technique. If you grew up in Texas and you raise cattle, you know about what's called the screwworm, and that was where it was first tried, where they actually irradiated males. They raised a lot of screwworm males and they irradiated them and then released them by the millions. And it worked for a very long time. And it worked because the screwworm female mates one time and then will not mate again. But then so here's what insects do. And so eventually there was a subpopulation where females would mate multiple times. And the system sort of started to break down a little bit. But
interesting.
That's what insects do. They're really very adaptable.
Right. If anyone, we have one more question in the chat, and if anyone, I know it's getting a little late, so thanks everyone for staying with us. If you have a question and you want to ask it yourself, feel free to unmute yourself and go ahead. And our last question in the chat was, is there anything new on BMSB, which I had to look up with the...
Oh, marmorated stink bug.
Yeah.
There are, again, yes, there are some parasitoids coming. That's the only thing I know about them in terms of one thing we haven't, has anybody had a serious problem? I mean, I have them around the house. My wife hates them. And I kill them as fast as I see them. But I haven't they haven't been a super problem yet, but the potential is really there. And so I do know that there's a number of parasitoids coming. I think there's one or two already being tested in the field. So we're getting close. And I think not just parasitoids, but also predators. And I know there's some native predators that will also, some other hemipteran predators that will go after them as well.
Okay, thank you for that. And someone mentioned that they had problems with the southern green stink bug on various kinds of beans.
Oh, yeah. Yeah, that one, yeah. That's a problem. It is. And there are some predators that will do it. You can buy predators for them. There is actually it looks like a stink bug that will go after the southern green stink bug. Yeah. I've not seen super numbers on them, but they can be pretty destructive. Yeah.
I had a question about lacewings because I know they are native here, the green and the brown ones, I believe.
Right.
I've never seen any myself, and I know you can order them. Actually, I was trying to remember why I knew Arbico Organics because I have their catalog and I was looking at the
Mhm.
ordering the lacewings. So what's your opinion on that?
Oh, they're great. They're wonderful. Great for aphids. There is I used to, those of you who know Nash Huber, what Nash used to do, he would put it into like a sugar solution. He would take the eggs and put it in a sugar solution, and he would just spray his fields with lacewings. And he would get just beautiful beautiful spread of larvae everywhere, of lacewing larvae. And they're great. They're very effective. You don't even, and you don't have to do that. But you do get, you get the eggs and then just put them out. They do work. I like them a lot.
Yeah.
For aphids, there is a midge. There is a midge that really works well. So if you really have an aphid problem, and I'm sorry, I can't remember the details on this midge, but it doesn't fly away as much. So it will establish here. And that's really great.
Is that a native insect?
I don't know if it's native or not. Can't answer that. Sorry, I don't know.
I'd be curious, yeah.
Yeah, but Arabco has that. It just look for the midge, and what it does is it's a maggot that feeds on it, and it sneaks up on the aphid and bites the aphid in the leg and feeds off it and just punctures a hole in it and just takes all the juices out of the aphid.
That's interesting.
Yeah, and so what you'll see is the aphid hanging from the leaf, just hanging from one leg from the leaf and dead. That's how you know you have them. And so they're here. They are here already. So they're in the, whether they're native or not, I don't know.
Right. Any other questions? I think we've gone through all the ones that were posted in the chat.
Okay. I was supposed to also do the murder hornet. Sorry, I forgot to put some things. I had some slides on that one. But
Oh,
anyway,
that's okay.
there's a lot of work going on that. That one is, I wouldn't be frightened of it. If you have honeybees, what I would recommend you doing, because this murder hornet is so big, is get some screening over your entrance that the honeybee can fly through, and the murder hornet will not be able to fly through, and you should be fine. They can't get in there, but if they get into your hive, they will wipe it out.
Yes, and I found it quite interesting that the honeybees that are in Japan have evolved to be able to kill murder hornets.
If it becomes a problem, we can start raising Japanese honeybees.
Exactly. Okay, well, if there are no other questions. Sorry, Deb, go ahead.
No, I was just gonna say thank you to David and thanks for.
Oh, well, thank you. It was fun.
Yeah,
thank you. I appreciate that.
Yes, very informative. Thank you so much.
Yeah, my pleasure. Happy to do it.